Hi everyone, my name is Fersen Leva.
And I'm Sandy Gramajo.
Today we will be presenting a preliminary research study that was titled Protease Activator Receptor 2 Agonist Increases Cell Proliferation and Invasion of Human Pancreatic Cancer Cells. This study was published by a team of Chinese researchers who are named on this slide. Before we begin to explain this study, Sandy will go over what a protease activator receptor 2 is, and some background information about the different components of this study.
What is a protease activated receptor 2? Protease activated receptor 2, also known as PAR2, is a G protein coupled receptor found in the cell membrane of various cells. And PAR2 expression has been associated with tumor proliferation, invasion, and metastasis in some gastrointestinal cancers, including esophageal, gastric, liver, and colorectal cancer. Normal pancreatic cells and pancreatic cells secrete trypsinogen. Trypsinogen becomes trypsin when it is activated, and trypsin is also an endogenous PAR2 agonist, but the cancer-promoting mechanism between the two hasn't been elucidated. In this study, pancreatic cancer cell line SW1990 was treated with trypsin, the PAR2 activating peptide, serine, leucine, isoleucine, glycine, lysine, valine, and the antagonist of PAR2 peptide, valine, lysine, glycine, isoleucine, leucine, and serine, to determine how these treatments affect PAR2 receptor expression. Moreover, it was found that PAR2 protein is expressed mainly in the cell membrane and cytoplasm of SW1990. To determine the expression of PAR2 protein in SW1990 cells, the researchers used immunocytochemical techniques, which included PAR2 polyclonal antibody, which was added, and then the cells were incubated overnight at 4 degrees Celsius. After that, a biotin labeled secondary antibody was added, and the cells were incubated again at 37 degrees Celsius for 10 minutes. Following that incubation period, horseradish peroxidase labeled streptomycin avidin complex was added, and the cells were incubated again for 10 minutes at 37 degrees Celsius, followed by staining with diaminobenzidine. When observed under the microscope, PAR2 protein expression should be shown by brown-yellow staining, but the image found in the article did not provide an accurate display of the image, and this color inconsistency might be related to the source of the article and not the image taken by the researchers. What can be seen is that the cells in the top image expressing PAR2 are darker than the control cells on the bottom image, which are not stained, and these results indicate that PAR2 is expressed mainly in the cell membrane and cytoplasm of SW1990 cells. In this study, one of the questions the researchers wanted to address is whether receptor agonists can affect PAR2 protein expression and pancreatic cancer cell proliferation. Another question the researchers wanted to answer is whether PAR2 activation can lead to pancreatic cancer cell invasion and metastasis. First, I will explain the effects of the different treatments on PAR2 mRNA expression shortly.
In order for the researchers to figure out if the different treatments affected the mRNA expression of PAR2, they collected mRNA with Triazole reagent and used reverse transcription polymerase chain reaction, also known as RT-PCR, after the SW1990 cells were treated. Also, beta-actin was used in the internal control for comparison of mRNA expression. This means that the PCR products were scanned by a gel automatic imaging system that expressed the values as a ratio of the absorbance of the detected band to the absorbance of the beta-actin band. The results of these procedures can be seen in Figure 2. On the top, the agarose gel that was scanned for PCR products shows the expression levels of PAR2 mRNA after the SW1990 cells received their respective treatments. The trypsin and SLI-GKV treatments caused a higher band absorbance to be seen in the third and fourth band compared to the control and VKGILS treatments seen in the first and second band. Again, these absorbance levels were compared to the beta-actin absorbance bands that corresponded to each treatment of the cells. On the bottom, the absorbance ratios between the part 2 and beta-actin bands were used to create this bar graph. The cells that were in the control groups, or VKGILS treatment group, displayed similar part 2 mRNA expression with values of 0.391 plus or minus 0.022 and 0.412 plus or minus 0.036 respectively. The cells that received trypsin or SLI-GKV treatment expressed more part 2 mRNA levels compared to the control group, with values of 0.645 plus or minus 0.038 and 0.612 plus or minus 0.042 respectively. Both were statistically significant based on p-values symbolized by the asterisks seen above the bars, where 2 asterisks means that the p-value was less than 0.01 and 1 asterisk meaning that the p-value was less than 0.05. This evidence suggests that part 2 is in fact present within these cells and that endogenous trypsin or other part 2 agonists can activate these receptors, which can increase the part 2 mRNA expression levels of SW1990 cells. Thus, if part 2 activation by endogenous pancreatic agonists like trypsin can upregulate gene expression, then this may facilitate pancreatic cancer cell proliferation. Sandy will now talk about how the researchers evaluated SW1990 cell proliferation that was affected by the different part 2 treatments.
The MTT assay was carried out to evaluate SW1990 cell proliferation after the treatments were applied. The MTT assay is used to measure cell proliferation rates, and the way it works is that the MTT solution is reduced by metabolically active cells, and the resulting purple formazan can be solubilized and quantified by spectrophotometric means. The cells were suspended and incubated in a 96-well plate with a density of 6 times 10 to the 4th per milliliter following culture at 37 degrees Celsius for 24 hours. And then the medium was replaced with a serum-free medium, and cell suspension was continued to culture for 24 hours. Following that, a total of 20 microliters MTT solution was added in each well after 48 hours of culture. 4 hours later, the culture medium was removed and dimethyl sulfoxide was added to each well prior to oscillation to allow the crystals to dissolve. After that, an absorbance value for each well at 490 nanometer wavelength was determined by the enzyme mark instrument. So in figure 3, it was found that the peptide VKGILS did not affect cell proliferation significantly when compared with the control group, indicated by a p-value of greater than 0.05. But trypsin and SLI-GKV increased the proliferation of SW1990 significantly in a concentration-dependent manner, indicated by a p-value of less than 0.05. As shown in the graphs, greater concentrations of trypsin and SLI-GKV groups enhance cell proliferation, and these results imply that part 2 agonist activation could promote pancreatic cancer cell proliferation. Now that we have seen how part 2 agonist activation could promote pancreatic cancer cell proliferation, first I will talk about how agonists affect the invasion and migration of SW1990 cells.
The researchers wanted to figure out if the different treatments on the SW1990 cells affected their ability to degrade and penetrate the extracellular matrix microenvironment. They decided to carry out invasion and migration assays with the Transwell chamber method that contained a degradable and permeable matrigel matrix, which essentially allows cells to move around the different layers of the gel matrix. The results of these assays are seen in Figure 4, where migration is shown as the gray bars and invasion as the black bars. The number of cells that accomplish these activities in the matrigel matrix layers are accounted for on the y-axis, and how the different treatments affected the number of able cells is attributed on the x-axis. More SW1990 cells that were treated with trypsin or SLI-GKV were able to degrade and invade through the matrigel matrix, with values recorded at 100.8 plus or minus 12.9 and 89.6 plus or minus 10.9 respectively, compared to the VK-GILS and control groups that had values recorded at 50.1 plus or minus 7.5 and 43.3 plus or minus 9.2 respectively. More SW1990 cells that were treated with trypsin or SLI-GKV were able to readily penetrate and migrate around the matrigel matrix with values of 81.5 plus 8.1 and 71.7 plus or minus 5.2 respectively, compared to the VK-GILS and control groups with values of 32.2 plus or minus 7.8 and 28.3 plus or minus 5.6 respectively. These invasion and migration results were statistically significant based on p-values symbolized by asterisks, where 2 asterisks meant a p-value less than 0.01 and 1 asterisk meant a p-value less than 0.05 compared to the control. This data shows how pancreatic endogenous agonist activation of part 2 could be related to pancreatic cancer cell invasion and metastasis, which are key characteristics of malignant tumors. Furthermore, in order for metastasis to occur, there must be degradation of the basement membrane and extracellular matrix, which allows tumor cells to invade the circulatory system or a neighboring organ. This damage of the microenvironment is an important sign of an invasive malignant tumor. Sandy will now further explain how this process was evaluated by the researchers through the different treatments of the SW1990 cells.
Researchers wanted to investigate how the different part 2 agonists affected the mRNA expression and enzymatic activity of matrix metalloproteinase 2 and matrix metalloproteinase 9, two known matrix metalloproteinases. Matrix metalloproteinases, or MMPs, help with the degradation of the extracellular matrix and the basal lamina of endothelial cells. And because they degrade these structures, the tumor cells are able to invade the tissue layers so that they can reach a blood vessel and invade the circulatory system until they can find a distant location where they can proliferate. The reason the researchers were interested in these specific enzymes is because part 2 is known to signal the production of MMP2 and MMP9 during colon cancer invasion and metastasis. So the researchers used RT-PCR from the gel observed in Figure 5 to evaluate the mRNA expression levels of MMP2 and MMP9. From the graph in Figure 5, it can be observed that MMP9 mRNA expression didn't change in any group treatment, as indicated by a value greater than 0.05. But MMP2 mRNA expression increased significantly in trypsin and the peptide SLI-GKV because it was recorded to have a value of 0.921 plus or minus 0.032 and 0.712 plus or minus 0.021 respectively, when compared to the values of the control and the peptide VK-GILS, which were 0.310 plus or minus 0.038 and 0.378 plus or minus 0.029 respectively. The researchers then used gelatin cymography to measure the change of MMP2 and MMP9 enzymatic activity. And gelatin cymography is a method of detecting proteolytic enzymes capable of degrading gelatin, and it is useful for the assessment of MMP2 and MMP9. Figure 6 shows how the agarose gel was stained by the presence of MMP2 or MMP9 after incubation. The optical density of these strips were analyzed and used to generate the bar graphs that compare the enzymatic activity of these MMPs. So the results show that MMP9 enzymatic activity wasn't affected by any treatment, but MMP2 activity was significantly higher in trypsin and SLI-GKV groups, indicated by optical density values of 75.6 plus or minus 6.1 and 60.4 plus or minus 4.6 respectively, when compared to the control value of 44.0 plus or minus 4.21 and the VK-GILS value at 39.3 plus or minus 5.2. This evidence indicates that agonist activation of part 2 induces MMP2 gene upregulation and enzymatic activity, which then may facilitate pancreatic cancer cell invasion and metastasis.
In summary, the human pancreatic cancer cell line SW1990 was found to express part 2 mRNA, which produces the part 2 proteins that are found in the cytoplasm and cell membrane. This evidence might be associated with the pancreatic cancer cells found within patients that have pancreatic cancer. Additionally, when receptor agonists like endogenous trypsin were used, they increased the part 2 mRNA expression of SW1990 cells, which could relate to part 2 activating abilities of trypsin within pancreatic cancer patients. It was also found that agonist activation of part 2 promoted SW1990 cell proliferation, which might be an effect from the part 2 mRNA expression increase just discussed. Furthermore, the invasion and migration abilities of SW1990 cells was enhanced by part 2 agonists, especially trypsin. This implies that trypsin may facilitate metastasis of pancreatic cancer cells within patients. Backing this implication is the data that recorded only MMP2 mRNA and enzymatic activity to increase in the presence of part 2 agonists. This finding might be related to the actual metastatic mechanism of endogenous MMP2 produced after trypsin activates part 2 in pancreatic cancer cells. All of these findings may elucidate a possible part 2 MMP2 signal transduction pathway that has the potential of being a target for the prevention and treatment of pancreatic cancer. Since this was a preliminary study, further research is needed to fully understand the hypothesized pathway. This concludes our presentation. Here are the 6 references we used to research the topic of this study. Thank you for your attention, and we hope this was informative.